In chapter 3, we'll talk about protein structure and protein function and how these amino acids, these long chains of amino acids, self-assemble and interact with each other based on intermolecular forces. So these are all things that we'll talk about in this chapter lecture. And this is just an image of a RNA binding protein. So it's a poly A binding protein that has multiple components. You can see that it has different types of domains or different types of characteristics that are shown in these ribbon structures. And over here is kind of another kind of line structure generated that shows you how these things are bonded to the poly A RNA. And these are just all of the amino acids that are attaching to the different components of the RNA. So these are all the things that we can do. We can look at structures not only that proteins are making, but how they interact with other things within our cells. And that's kind of the basis for this. And the understanding of these can help us understand what the function of that protein is. So this has a structure that the structure is really important for its ability to interact with RNA, for example. In the previous chapter, we talked about some of the macromolecules, the common small molecules in cells. Amino acids, we need at least 20 of these, and that are going to be components of our proteins in our cells. We have aromatic bases, adenine, cytosine, guanine, thymine, and uracil. We have different types of sugars based on their interaction in terms of where they're interacting off the chain. Ribose, so is a component of RNA. Deoxyribose, component of DNA. Glucose is a component of energy and metabolism. And of course, we can take glucose and make these other types as well and interconvert those through our metabolic processes. Lipids are fatty acids and components of long hydrocarbons. And cholesterol is another component derived from fatty acids that is necessary for normal cellular, extracellular membrane fluidics. There are different classes of proteins. I want to walk through those briefly just to give an idea of all of the different types of things that they do, and they're involved in nearly every aspect of the cell function, and they're all over the place. So there are enzymes, for example, that are really the function is to catalyze and the breaking down and building up of different macromolecules such as lipids and cholesterol, as well as molecules that are involved in energy, carbohydrates and pyruvate and all of the components that go through the mitochondria and the citric acid cycle and the electron transport chain and all of that. But there are other different types of enzymes for all sorts of purposes. We don't focus in this class very much on the enzyme component of proteins. We focus on some of the others a little bit more, and the enzyme part of this is more described in the metabolic biochemistry course. So all cells have certain types of structures, and these are the main components to give those cells their unique shapes that might have different functions. We have actin, microtubules, and intermediate filaments. So we have structural proteins just to build the cell and make it look like what it does. And we'll talk more about those in later chapters and talk about how those are involved and where we find them and what their specific purpose in the structure is. We also have transport proteins, and they are carrier molecules, so they can carry small compounds such as hemoglobin or hemes or other types of metals or other types of metabolites that are required for whatever structural functional purpose within the cells. We also have motor proteins, and there are different types of chains, for example, that are part of the structural component that we were talking about earlier. And then there are things that kind of use those structural proteins as a highway for moving up and down. So if you think about an axon and you think about how things might need to move from the nucleus out to the periphery or out from the periphery back to the nucleus, and those things, there are different types of proteins that crawl along those highways to help with active transport, help move things back and forth within the different domains of the cell. So we have a myosin heavy chain, which is a component of the structural functional part of a cell. And then we have kinesin for microtubule transport and dynein for cilia and flagellar movement. So different types of movement, and there are different, if you're talking about muscle movement, we're talking about myosin. If you're talking about intercellular movement, we're talking about the kinesin, for example. Some proteins, their function is just to hold onto certain components or certain metabolites. Ovalbumin, for example, is a protein that's expressed and put into eggs, for example, and they just are, the whole purpose of it is just to have all of the amino acids within it so they can be broken down during development to use as building blocks for building the embryo. Other examples of this are ferritin protein, which stores iron in the liver, and there's also casein, which stores amino acids in milk. So that's why milk is such a good source of proteins and the building blocks for building muscle. In terms of, that's why we use that. And as my younger son would attest, he's really into drinking a lot of milk, and that's a good source for all of the building blocks for proteins for his body. There are also signaling proteins, and not only signaling molecules that come in and bind to receptors, but then groups and sets of other proteins within that carry out those signals from one cell to another and into the nucleus, that then can activate transcription factors and generate genes that can then be made into proteins and then go out and signal other things as well. So we have hormones, growth factor signaling. Insulin activates enzymatic pathways for changing your metabolic processes. We have different types of growth factors. Netrin, for example, influences axonal growth. So an axon, if we want to move towards one cell or another, it would get that signal. Different growth factors and all sorts of different things that will change the processing and the metabolic function of our cells. And they're really important sets of proteins that we'll talk about in later chapters. I think along with that past slide, we have the signaling proteins, and typically there's going to be some type of receptor or something that is going to receive a signal on the outside. We have these in our eyes. Rhodopsin detects light, and then that changes the structure, and then that influences a signal transduction mechanism to signal to our brain that we are seeing certain wavelengths of light. Acetylcholine receptors, that helps to change muscle contraction. The insulin receptor I mentioned before, changing our metabolic processes. And there's all sorts of other ones that will go into some of them, not obviously all of them, as there are a tremendous number. There are a lot of gene regulatory control proteins that are going to bind DNA, change DNA, allow the expression of different types of mRNA off of the DNA, things that inhibit gene expression off of the DNA. So looking at genetic control, looking at developmental control, looking at cellular function control. So these are all different types of regulatory proteins. There are all sorts of those. And finally, there are some special purpose proteins. If you are looking, for example, here's an antifreeze protein that is involved in Arctic fish protection. It protects the blood from freezing in Arctic waters. You've probably heard of green fluorescent protein that green jellyfish had and use, and then we've co-opted that protein to look for and to use in different types of biochemical assays. There's a really interesting protein, monelin. It has a really sweet taste, and it's found in what's called the serendipity berry from Africa, and that's shown over here on the right. It's kind of an interesting little berry. It denatures at really high temperature, but it's a very sweet flavor, but it's a protein. It's not sugar. It's not glucose or any type of glucose type of protein, it's not a carbohydrate. It's a protein. So it is very sweet tasting, but it's actually made up of amino acids, which is pretty neat. Typical amino acid structure, we're going to have an amino group. It's attached to a carbon with a side chain and a hydrogen, and then that central carbon is going to be attached to a carbonyl group or carboxylic acid. So this is a carboxylic acid functional group. And then when it attaches this, we'll talk about the different types of structures that are formed here. So this is the amino acid. So the amino carboxylic acid, that's kind of that component over here. There are really only 20 kinds of amino acids are used in human protein synthesis. Some contain additional amino acids, and usually as a result of different modifications after the protein has been synthesized. And there are no two different proteins have the same amino acid sequence. So you're not going to have the same sequence and then it's going to just be a different protein. So each protein would have a different unique amino acid sequence. So here are some of the functional groups, and it's based off of I know we kind of wanted to get away from hydrophobic, but really lipophilic is probably a better word, but liking fats. And so these are non-polar amino acids, which means that they tend to not interact with water. So glycine is the simplest, where it just has another hydrogen off. This also, by the way, now is not a chiral center because it is attached to two things that are the same. Alanine, this is a chiral center. So a lot of these, as I can show you in this previous slide, we have an alpha carbon, that's the central carbon, attached to a hydrogen, attached to the carbonyl group or the carboxylic acid group and the amino group. And here we see the side chains that are going to be different. So we are going to have, there are possibilities for different types of amino acids, but in our bodies, in life on Earth, we typically only use the one type of amino acid that's utilized within protein synthesis. So we don't use both sets, we only use one set of those. We have valine over here, which has 3 carbons in a row. Leucine has the 4 with kind of the V at the end. Isoleucine is like that. Methionine has a sulfur group. Phenylalanine, we have an aromatic ring at the bottom. Tryptophan, a big ring, and also an amine group attached in another ring. And then proline, this is an interesting one here where it actually forms a ring structure, and this also causes a bend in protein structures. So it causes this kind of hairpin turn. It's also something that gives us digestive issues for certain people. There's a lot of, if you think about gluten-free things, gluten is a protein and it has a tremendous number of proline amino acids in it. And sometimes our body don't like that structure, and sometimes we have difficulties digesting this protein because of the multiple groups of proline that cause different irregular odd shapes, and sometimes cause problems with digestion or sometimes even autoimmune responses. This set of amino acids, hydrophilic, so liking water. We have serine, which has a alcohol functional group, and then threonine, which also has an alcohol functional group with an extra carbon attached. Cysteine is an analog, which means that instead of an oxygen, we have a sulfhydryl group. Cysteine here. Tyrosine, we have a big aromatic group with a hydroxide group. Asparagine, we have this amide functional group on the end. And glutamine, again, an amide, but we've added an extra carbon within that chain. So asparagine and glutamine are very similar, but one's a little longer than the other. If we go down to the hydrophilic, which again means that, and then they're also charged, so there's a charged state. So these are uncharged up here, so typically will not have any charge associated. But aspartate and glutamate, typically under physiological pH around 7, these are going to be acidic, which means that they'll lose their hydrogen at the end of that carboxylic acid functional group. Lysine would be typically, these are basics, they'll be positively charged. We'll have an amine group and another amine group and then another amine group. So these tend to be basic compounds and take on positive charges. So here again are the 20 amino acids and the 3-letter abbreviations and then the 1-letter abbreviation. So you won't have to memorize this. It's not a bad idea to have a general idea of what you're talking about. I think the hardest ones usually lysine and leucine, because they both theoretically could start with an L. And then most of the other ones kind of make sense. I think glutamate's kind of a little odd. Glutamine and glutamate sometimes get difficult to differentiate. But typically a lot of these kind of make sense in terms of what the name is based on what the letter is. But you don't have to memorize these. If you need, I will probably have a list or show you what they are somewhere if it would be on an exam or a quiz, but nothing that you should have to memorize. But the more you know this, the easier things get in terms of having conversations or understanding what they are. You can move a little faster in your homework or your quizzes the more that you practice with these. So these are the individual units, and we have the amine group, the central carbon, and the carboxylic acid component. And what we're going to do is we're going to actually take these ends and we're going to basically dehydrate it. So we take this oxygen and 2 hydrogen, and we're going to form a water. And at the same time, this nitrogen and carbon are going to make a covalent bond with each other. And so then you can see that water is excluded, and then we've made a peptide bond, which also is an amide, A-M-I-D-E. It's an amide functional group. So this is the amide functional group is also referred to as a peptide bond. So now we go from amino acids to a peptide, which is a multi-group amino acid. Here's another cartoon of a peptide, so a short sequence of amino acids, and we have methionine here. So we have the side chain coming down, and then they alternate going down, going up, going down, going up in this cartoon. And then we can see these side chains here. Aspartic acid's charged. Tyrosine is going to be somewhat polar group with that hydroxide here. And we can see that we get NCC, 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 NCC. This N side, this is called the N terminus. So by convention, we typically would draw the N side first. So N comes first, and then the C terminus is the carboxylic acid terminus, or C terminus end. And this is also going to be by convention if we go 5' to 3' when we're looking at making mRNAs, and then the mRNAs being translated into proteins. It's N to C, 5 to 3. That's typically the convention that's used to set up our sequences, regardless of if we're talking about DNA, RNA, protein. That's kind of, at least they rhyme, so we can kind of keep them set up the correct way. So that's important because your 5' sequence to your C' sequence, if you're reading through that reading frame, that first set of 3 different types of nucleic acid are going to be what encode for the different types of the amino acid sequence. Our case study for this chapter focuses on this idea that proteins fold into low energy conformations. So when you're synthesizing your protein, you're going to create all these side chains that are going to kind of come off different directions. And what your protein is going to do is basically self-assemble. So it's going to hide the things that are kind of lipophilic, right? So the things that kind of like lipid better, and then the hydrophilic things are going to kind of move themselves towards the outside. So as this is synthesizing, those components that are happier by fats are going to interact with each other, and those things that want to be interacting with water or other polar compounds are going to sort themselves out to the outside. So this is an interesting component here where there's these bonds that are made in water are really strong, and they're going to exclude the incorporation of these kind of green ones on the outside because they'd rather make those connections with the things that want to make those connections the most here. Okay, so a bit of a learning check. I just want you to stop. I want you to get something to write this down on, and then I'm gonna have you bring this. We'll have this. You bring this to class, and or answer a little worksheet thing about this. I'd like you to explain why cysteine residues with free sulfhydryl groups tend to be localized on the exterior of a protein molecule, opposed to those involved in disulfide bonds where they're more likely to be buried in the interior of the molecule. So take a moment and write this answer down, and then once you're ready to go, then you can resume the video. Okay, so back to protein structure and this idea that they can self-organize. One of the reasons that we know this can work is because certain proteins, if you purify a protein, isolate it from the cells, you lyse the cells, you pull out the protein, you would purify it with some type of chromatography or some type of pull down technique. And if you were to take that and expose it to high concentrations of urea, so that's shown here. We have basically an amide functional group with another amine on the side. And this is a common component of urine, so urea, and it's kind of a breakdown product of proteins. And what happens is, because this has a similar structure as the backbone of a protein, you can see that amide functional group is kind of like kind of a peptide-like bond, but it's a lot shorter. So what happens is this can intercalate and get into all these little cracks and crevices. It's very soluble within that folded protein structure, and it can break it apart. It interacts and it allows that protein to denature and unfold in solution, because this is water soluble. So it breaks apart all of those bonds and all of those interactions. And if you remove it, so then this is water soluble, and if you kind of dilute that out or get rid of it, then what happens is that will refold back into the native conformation, because again, it's based on that lower energy conformation. Now this doesn't always work, but for some simple proteins, you can get that to fold back, and that tells us something about the structure. And it will actually be a functional protein. So if it's an enzyme, it'll have the same enzymatic processes. If it's, it typically will look exactly the same, and if it looks the same, it should function in the same way. So these are really important concepts and things that the basis for that folded activity that we'll do with for the case study for this chapter looks at really the energetics of the system. It looks at using a mathematical program to say, okay, this is the most stable structure. It's the lowest energy, the most stable conformation, and this is probably close to what the actual folded protein looks like in real life as it's functioning in our cells. So the protein folding situation I showed you with the urea in that last slide, that can work for some proteins. Some proteins are a lot more complex and they have more components and parts that are coming together. And sometimes they need what are called chaperone proteins, so that they need help folding. But these things are all kind of expressed in the cell, and they can help bind certain components and bring other groups together and different functional groups together and help to correctly fold those proteins. Heat shock proteins, chaperone proteins, those are all help to protect and fold proteins under abnormal conditions where heat, for example, obviously heat shock would be help to denature proteins as well. If you have these heat shock proteins or chaperones, they can kind of go into this. There are two different types, some that bind the outside and bring them together, and then somewhere they'll kind of create kind of this barrier and keep other things away from it so it can fold in the conformation, lower energy conformation in a certain, in an environment that's conducive to that, and then it will release it and it can then go on and serve its purpose. This is a cartoon of a three-dimensional structure of a chaperone protein from E. coli. So it is a complex. You have the top and the bottom. There are multiple stacks of different types of rings and different components of proteins that are shown in different colors here, that where you could have the synthesized peptide come in here, fold, and then come out as a functional protein. So again, these are available for when and get more highly expressed if the conditions where the E. coli is living, for example, are a little more harsh or outside of standard conditions. And it allows for proteins to still fold normally and then come out and be functional so that the cell can serve its purpose. And protein folding is really important. So there are certain types of proteins that can misfold and that can cause cellular aggregates that can cause abnormal cellular function. So prions, for example, are something that where maybe you have your normal folding type and then it'll get flattened or changed the structure, and then that will bind to another normal thing, and then it'll cause conversion of normal proteins into abnormal fibrils or prions that will then stack up and cause cellular function and issues within the cell. So this is an example. If you have amyloid fibrils that can cause Alzheimer's or different types of prion diseases that can cause neurodegeneration and brain issues. So a big important part of this chapter is that all of these different things have different structures, right? Collagen is a long, big long chains of proteins. You might have something that has a different types of functional groups that are going to stick out and to interact with different groups. You have different proteins that are going to interact with DNA, for example. You're going to have all sorts of different things that are going to be recognized and received, and they all have different purposes and they all have different shapes. So that is the big thing, is the structure is going to tell us something about the function and what it's interacting with. Okay, here's another learning check. I want you to take some time to think about this and feel free to talk to other people about it and kind of come up with a solution for this idea or this question. So if we're looking at the following genetic mutation, so we have the DNA sequences shown here, and we have the normal cell line, and then we have an abnormal cell. And so we have a difference of, we go from a CTC to a CAC. And the CTC encodes for a glutamic acid, and a CAC encodes for a valine. So this is the normal structure, and this is the mutant structure. And the only thing that's different between these cells is this one point mutation. This causes, this allows us to have normal red blood cells that have a normal shape. And then this different DNA sequence, this causes a mutation that causes sickled red blood cells or elongated red blood cells. So I would like you to propose a hypothesis as to why this one change makes the cells look different. So just take a moment, write that down, and then use that to answer. We'll use this to answer the question. I'm going to check your that you answered these, and then that's it. So this is the first part of this lecture, and then what we're going to do next is we are going to look at this, we use this fold it game that we're going to, this is going to be, I'll give you the directions and the proper link to get to that. So for our biochemistry class, so we're going to really use a computer system that's kind of like set up like a game. So you're going to go through these processes to kind of understand protein folding, different types of proteins, how they fit together. And then we're going to use this to go into an actual primary literature article that used that to understand some of the protein structures and use that to solve some problems in that field. So this is the first part, and then answer those questions, and then what we'll do is we'll work through the case, and then we'll go through some of these other slides as well.